We get experimental science. We're curious, non-judgmental.
So it is a snake, and it's named after Harrison Ford. Yes, that was not potentially the most creative name, but it is in fact called Harrison Fordy. So there are naming conventions for genetics that is dependent on the community that you're working in. So for fruit flies, it's pretty open. As long as it hasn't been described in another organism, you can name it whatever you want to. If it has been described in another organism, you have to make it a derivative of that name. So if it's a toll receptor in one organism and then you find it in a fly, then maybe you name it toll-like receptor, something along those lines. What's neat is that these papers on new species very much run in the same vein pretty much every time you see them nowadays. It's very unique to have a lot of depth in these papers. The reason I say that is because a lot of times it's sequencing, physical imaging, and that's that. There would be more fun ways to be able to analyze this kind of data, but given how the publications work, and how the finances are, sometimes that's all you're able to get is just the basic. We sequenced, we did physical comparisons, and that's that. And so that's a little component there to think about. They do have the distribution map, which I do very much enjoy. So some of the papers that we looked at for new species don't end up having distribution maps, which to me was very frustrating because you have no idea where they're coming from. This is their distribution, and if we look at the map, it's the snake is located in Peru, and they actually give a nice geographic topological map of where these animals were identified. And they actually looked in this national park, and then here in these thumbtacks, these yellow thumbtacks are the 3 areas that they identified the snake. That they did multiple sequencing samples, which was really good on their part to do because they further proved that yes, in fact, this is a unique snake species. They didn't just use one individual that they were like, okay, this is different. The reason it's bad to use just one individual to make a comment on the entire genus is because you don't know if it's a mutation in that one particular individual that makes them weird, or in fact they are a separate species. And so that's an important distinction to be able to make. That's why I'm really glad that they took 3 samples to sequence. They also took 3 samples for preservation and measurements as well to get the detailing of them. You don't know if you sequence one individual if that's a mutant, if that's the norm, if that is a different species. It's always good to have more samples. And so I'm glad that they actually went in and got multiple samples for this particular species. What's neat on this paper is that they're super, super high resolution for the images. So that's why you saw it takes a bit of time to load, but it's also something that we can really zoom in on. So what's unique about these snakes is these particular skin folds that you see. That's common in the genus, located right here. So these are like folded regions of skin that are like just clustered together. These 2 little lines here. And it's just, mechanism-wise, it's honestly not great on proving what exactly the point of these are. The hypothesis is for feeding behavior, but is it really proven? No. Remember, there's been no genetics done on these animals. You can't yet prove what the purpose of any of these features are, so it's suggestive. That's all we can say about that front. And see, here are the multiple folds, like these little rails that are going through. It almost looks like individual train tracks. Those additional regions are from the snake. And so one thing that they were doing is pointing out regions of their body. These particular folds that they're highlighting are unique features of these snakes. So if you're looking at the runner of this animal, it has these 3 like almost railroad-like body part features, like of they're running down on their skin. And that's supposedly a unique part of these animals that they can be identified by. Members of this genus also have that. That's the suspicion, Sparky, that it helps with digestion and it's like musculature that allows them to crush what they've eaten. But these kinds of things you can't ever prove until you can do genetics on these animals. You can't actually there's no validation of these hypotheses. It's just, we think that's what it is. Whether or not it is, we'll only find out if we do the genetic tricks on them. So they actually did a couple of images here in the study of the living animals. So this is the underside view. That's why it's the different color. So that is the underside of the snake. the ventral side, and there is a dorsal side and a side view as well. So this is a zoom in of the head, just so you can see the side view, the dorsal view, and the ventral view. Just again, high-res imaging, lots of different measurements that they did, and they do this to compare against other existing measurements that have been done on the other suspected species. This is the male holotype as well, so you can see the male is a little bit lighter in color than the female. They do sacrifice 2 samples here, a male and a female, that they're able to do measurements on. The reason that they do that is if you were to find a dead sample, it wouldn't have the same volumetric measurements as the living would have. So instead you preserve it in a living state so it doesn't shrink or, shear, or any kind of like weird effects wouldn't be happening to it. Instead, again, it's that living one, you preserve it, usually ethanol or methanol preservation, and that's when you do your measurements on. Then when someone goes back and looks at it and they do the same measurements, let's say that you find a living snake of what you think may be different than Harrison 40, you want to do the measurements on the original to double check. If you were doing the measurements on a dead one, it could have different like widths and lengths as a function of it had dried out, and you might be misinterpreting the data, and so that's why they do it like that. Here is habitat images. These are the locations where they were caught. So again, this is a Peru like high latitude mountain range, and then here like in the underbrush is where they actually identified them. And even here in the circle, you can see that there is a snake in fact inside. So there is a little head sticking out, and they're able to. It was cool that they actually brought in an image where you can see the snake being identified. There's the snake right there, and there's the rest of its body. So it's a nice little scale as well for the size plants next to it. And there we have the little snake head. What's neat is they have an entire section in their paper called for the etymology of why they named it what they named. They say, we dedicate the species to Harrison Ford, actor and conservationist, in recognition for Conservation International and his voice for nature, i.e. nature is speaking, Harrison Ford is the ocean. So they did not name him after the fact of Indiana Jones, as the popular media is saying, but in fact because of his work as a conservation advocate.